So moving now to genetic targeting in pancreas adenocarcinoma, we've known for a while that there's a subset of patients who have defects in DNA repair and may benefit from selected targeting. And the concept behind this in BRCA deficient cells is the concept of synthetic lethality, where individuals with BRCA-driven malignancies have an ineffective ability to repair DNA damage, and alternative mechanisms which are much more unstable and lead to cell death and depletion, lead to potentially higher therapeutic advantage in BRCA deficient cells for some platinum and potentially PARP inhibitor strategies. This is an example of a study. This was a prospective multi-center small trial in 2 cohorts of patients, those who had a germline BRCA mutation and those that did not. For those that did have a germline BRCA mutation, frontline therapy with platinum and PARP combined, we saw significant responses. We saw no objective responses in the non-BRCA mutation group. Similarly, some of these responses were very durable, going on for a number of years. And again, in this small subset, and a caveat, to note that it was prospective and multi-center, we saw approximately a doubling of survival for patients who received platinum-based therapy as their initial treatment strategy compared to those that did not have a germline BRCA mutation. And it's sort of unclear how much of this is predictive versus prognostic effects, but our understanding is that it is primarily a predictive effect of platinum in the BRCA mutated subgroup. And that's led to a series of studies. This particular trial has just completed recruitment and will evaluate platinum and PARP inhibitor upfront, as opposed to platinum-based therapy on its own for germline BRCA and PALB2 mutated individuals with advanced pancreas cancer. And a very important study we hope will read out in 2019 evaluates a maintenance approach of using a PARP inhibitor following platinum stabilization for patients with, again, a germline mutated pancreas adenocarcinoma. And the study builds on themes of PARP inhibitors in ovary cancer and in breast cancer, where significant benefits have been identified, and we hope that this will be also seen in pancreas adenocarcinoma.